now, I would like to invite everyone to join our next session. It's a great pleasure to introduce our speaker, Dr. Scott Jackson. Please, everyone, help me to say a welcome to Dr. Scott Jackson. Dr. Jackson is currently the Georgia Research Alice and Eminent Scholar in Synthetic Biology at the University of Georgia, a position he assumed in 2024 after serving as a North America breeding product development leader at BioCrop Science. He holds a PhD and master's degree in plant genetics and plant breeding from the University of Wisconsin-Madison, as well as a postdoctoral training at the University of Minnesota. Throughout his career, Dr. Jackson has combined academic leadership and industry experience with previous roles as professor at Purdue University and the University of Georgia, director of the Center for Applied Genetic Technologies, and program officer at the National Science Foundation. He has also contributed to the scientific community as associated editor for leading journals such as the Plant Journal and Molecular Plant. Just a quick reminder, everyone, we will have a question and answer section after the lecture. Dr. Jackson, thank you very much for joining us today. The audience is all yours.
All right. Thank you, Bruno. Thank you, Gabrielle, for career path. I'm going to go into my career path shortly. Can you hear? Is this close enough? Is it on? Now is it on? All right. Did you hear me thank them already? I don't have to do that again. All right, perfect. If you thank them too much, then they get big heads. All right. Figure this thing out before we start. So first of all, it's a pleasure to be here. This is, I think, maybe my 6th or 7th visit to Brazil. Every visit has been wonderful. Don't tell any of my friends at the other places I've been to. This is by far the best visit so far. So thanks to everyone. Gabrielle talked about career paths not being linear. Mine's a circle, just because it worked out in the slide. And I thought it'd be useful for some of the students just to talk through what my career path has been like, because I'm actually very old now, much older than most of you. So I did as Bruno said, I did my PhD at the University of Wisconsin in Madison, and I did that in plant breeding. I worked on potato, and then I worked on cytogenetics of a number of species, including rice. I finished my PhD in 1999. Who here was born after 1999? I know there's more than those 3 or 4 hands. Yeah, thank you, Tiago. Probably all the students and postdocs in here were born after I got my PhD. That's when you know you're old. So as I said, I worked on cytogenetics at the end. I love chromosomes. I saw several posters with chromosomes, so that's always been my passion. I did a short postdoc at the University of Minnesota where I carried some of my rice research and started working on wild relatives. So I've always had a very deep interest in using, understanding and using wild relatives for crop improvement. In 2001, I went to Purdue University in Indiana for my first faculty position. And I was hired to work on a species that I'd actually never done anything with. I was hired to work on soybean genomics. This is 2001. The only thing I knew about soybean is that when you drove down the highway in the Midwest of the US, the tall stuff is corn and the short stuff is soybean. That is literally all I knew about soybean. And they took a chance on me and hired me to work on soybean genomics. I continued to work on rice. This is some of the wild relatives distribution of rice. We started a soybean genome sequencing project that was done almost a decade later. In 2011, I moved to University of Georgia, and I picked up some other legumes. I'll talk about some of the genomes that we've worked on, and picked up some more modeling and functional genomics activity at that point too. And this is where that sort of stepwise career path that Gabrielle was talking about takes a diversion, because then I went to industry. I had 18, 19 years in academia. Been a wonderful time, loved it, it was great. And the reason I went to industry at that point is really the title of this conference, Integrating Omics. My PhD was in plant breeding. I'd done 18, 19 years of generating omics data sets, and I really wanted to apply that to plant breeding. And that's somewhat difficult in an academic setting, especially at that time, just because of the cost, the scale. And when Bayer approached me in 2018 and asked if they'd be interested to come and develop a group that would focus on how do you apply these different omic technologies across a breeding pipeline, I was like, wow, this is what I've wanted to do. This is the chance to do it at scale. And so I said yes, and I went to industry for 5 years. Had a fantastic time. There's some Bayer people. I think they left to get interviewed, but Gabrielle's for Bayer. Just a wonderful time in industry. It was great. I thought that was my last career move. I'd retire from Bayer. But Georgia came calling again and asked me to come back and start a new institute. And I'll talk a little bit about what we're doing there, and we're calling it crop design. It's trying to bring all these new omic technologies, modeling tools, quantitative genetics, and everything together to really make an impact in how we do breeding in the future. So I know this is a plant breeding group, so I'm going to give you what I think are the major innovations in plant breeding. Most of you will disagree with what those major innovations might be, and I'll miss some that might be important for you. But if you think about breeding, it started as selective breeding, picking the best out of a population, even going back to domestication. So picking the ones and advancing the ones in the next generation where the seeds didn't fall on the ground. And that's how you got from shattering to non-shattering, a major innovation in domestication. And that continued all the way up through the 1920s and 1930s. It still continues today, selective breeding, although we've added markers and other technologies to it. It's random, it's slow, it's large, but it's successful. It's worked for hundreds of years, thousands of years. 1920s, 1930s, we had the innovation of hybrid breeding. This was mostly corn at that point. Now we have hybrid breeding in a number of different crops. Actually, this very picture was shown yesterday. But basically, you have two inbred blinds. You cross them, you get a progeny that's much better than either the parents. But you're still using selective breeding for the inbred selection. Now I'm skipping over a lot of innovations that happened over the past 70 years, haploid induction and things like that, to the 2000s, where it's really the culmination of all the molecular tools, the genomics, the quantitative models, a lot of development that happened in animal breeding, to the point that we're using genomic-assisted breeding, genomic selection, genomic prediction tools. Still, in many ways, it's selective breeding, but we're using a lot of advanced quantitative models on top of it. So it's less random, it's a little bit faster, more targeted population development, and reduced testing. And over here, what's not appearing is crop design, which is just bringing all the different tools together. And so you can see the impact. I mean, this graph has probably been shown more than just about anything at every plant breeding conference in the world. But looking at corn yields in the US going back to the 1860s, all the way to 1860 and 1930, they were pretty flat. They were selecting for different disease and different ear kernel characteristics, but they weren't really changing the yield curve. And that wasn't until the single cross and double cross hybrid started in the 1930s. You had Pioneer hybrid corn that came on board, and you see how the yield went through from 1930 to present. And I think this is a really amazing way to picture what's happened. So if you look at, this is US data, so if you look at 1930s, the number of acres planted and harvested, about 82 million. 2020s, we're still about the same number of acres planted and harvested every year in the US. But we have about 6 or 7-fold more production per acre because of hybrid technology in those intervening years. So we still have challenges ahead of us. So I'm going to talk a little bit about, 4 different things. I'm going to talk about genomics, how they developed, and some of the things that we've contributed to from my lab in crops. I'll talk a little bit about big data, a little bit about AI, and I'm not an AI expert, so if you want to ask deep questions about AI, Alex is sitting in here somewhere. You can ask him. There's other people that have much more expertise into AI. But I'll talk a little bit about how we're thinking about using it and what we're doing. And then I'll come to the end where I hope to tie these things together to talk about how we're thinking about crop design for our future and bringing disruption and innovation into plant breeding. So my focus, I mentioned that we worked on rice. We did a lot of work on rice over the years. I'm not going to talk about any of the rice work. I'm going to talk about some of the soybean work throughout this, or legume work throughout this talk. There's some probably Macintosh to PC conversion issues going on, but that's fine. So my lab focused a lot on legume genomics. We started working on the soybean genome in the early 2000s. That was finally done in 2010. The years all disappeared. We worked on a number of other legume genomes up until I left academia and went to industry through 2018. And you can see arrows where they are. We helped produce these. These were large international collaborations for the most part. Not like generating genomes today where it's an overnight run on a PacBio or something. This involved multiple countries. In the case of rice, every country put a flag on a chromosome. China's doing chromosome 1, Japan's doing this chromosome, and it's almost like claiming territory back in the colonial days, that this is my territory. No longer the case. But these are large, complex projects with many participating labs and countries. Of course, the legumes are very important. Soybeans are very important in Brazil. You're, I think, the largest producer of soybean now. You overtook the US a few years ago. Congratulations. But there's many reasons why the Fabaceae are important, from nitrogen fixation, a number of crops and forages that exist in those. So it's a very important agronomic or set of species for production. Let's see, I keep getting these buttons mixed up. So every time I worked on cytogenetics for my PhD, so I did a lot of work on polyploidy. We worked on potato, worked on other polyploids. And I've always liked the conceptualization of the chromosomes behind polyploidy. And we heard about the wheat talk yesterday from Catherine, so that's obviously allohexaploid. But polyploids is this recurring cycle that happens in plant evolution. If you go back lineages, there's multiple whole genome duplication events. Some of those are retained over time as polyploids, either as auto or allo. Others go through a process of diplization, where you start to lose duplicate genes and duplicate parts of chromosomes, and it comes back to some semblance of a diploid, and then it literally goes through this process over and over. And so when you start sequencing genomes, you can find the remnants of this history in these genomes. And in the case of soybean, when you take all the genes and align them and use a Ks on the x axis and density on the y axis, you can start to find these peaks, and these peaks can trace back to duplication events. So what you're doing is aligning paralogs, paralogous genes that come from duplication events, looking at the evolution over time, estimating using molecular clock divergence, and you can see that we can find a very recent one about 13 million years ago and an ancient one about 55 million years ago, and you can actually find evidence of these in related lineages before or after these splits. What that means is, of the roughly 45 to 50,000 genes in soybean, 75% are present in more than one copy, 54% are present in 2 copies. There's very few genes present in 1 copy. And when you do all the chromosomes, and this, every genome paper has a Circos plot, so this is our Circos plot. It's amazing that 20 years later they're still doing Circos plots for genomes, but there it is. But you can find extensive retained synteny of blocks of entire blocks of chromosomes that are retained in synteny on other chromosomes at the micro level, high level sequence conservation between these regions. And these come from that most recent duplication event. Of course, sequencing has gotten better over time, and this is from a paper from Marks et al. Just last year, the year before, I think, or 2021. And here what they're looking at, genomes that were published over time from 2008 to 2021. And on the y-axis is the contig length. And you can see the contig length keeps getting better and better and better until you get to after 2021. Basically, the contig is a chromosome arm or a full chromosome, which is amazing. It was difficult to imagine that back in 2006, 2007, 2008, that you'd have a sequence assembly where you had complete coverage or a complete contig assembly of an entire chromosome or chromosome arm. And you can see the shift to long-read sequencing, the blue circles versus the green circles. So there's been a shift as the contig lengths have gotten better, so the sequence reads have gotten longer. And of course, the cost has come down tremendously. These are rough estimates on what the costs were. People have different numbers for what the actual costs were. The Human Genome Project, which was conceptualized in the late 1980s, was finished in early 2000s, roughly about $2 billion. Plus or minus $1 billion, depending whose estimate you use, for one genome. But that was a that's like the moonshot from the 1960s, 1950s, 1960s for physics, for biology. That investment led to all sorts of technological innovation in sequencing and and computational analytics, and that really advanced the entire field of biology. The corn genome, the soybean genome, both took 8 to 10 years. Estimates are $60 to $100 million each, depending whose estimates you use and what numbers, plus or minus $60 to $100 million. And those genomes are not that much smaller than the human genome. So you can see that from 2 billion to 100 million. We participated in the peanut genome a few years later. That took about 8 years. It shouldn't have taken that long, but it took about 8 years, about $6 million. Now you can do an entire genome for less than $1,000 overnight. So technology is rapidly advanced. All right, there's a lot more genomics in crops. That's all I'm going to talk about. Talk about big data. So what is big data? There's concepts of what they call the three V's. This is from Doug Laney, some work that he proposed back in the 2000s. There's the volume, the amount of data, the velocity. That's how the rapidity or the amount, the pace at which it comes. And then the variety. And this is actually an important concept in biology, the variety. And the reason I harp on that a little bit is when I was at Bayer, we started hiring some physicists because they understood big data, and we were starting to generate what we thought was big data in breeding pipelines. And from an Illumina NovaSeq, you generate 8 to 16 terabytes. You think that's big data. Physicists generate petabytes per second, depending on what they're doing. The large colliders, like the Hadron Collider, petabytes per second. These large camera arrays for deep space imaging, petabytes per second. So our concept of what big data is dramatically different than what physics is. But the difference is that physics data tends to be very structured and not very complex. Whereas biological data, you have sequence data, you have RNA-seq data, you have all the environmental factors, tends to be very complex and very heterogeneous as compared to physics data, and much more complex. So there's differences in perception and how we perceive big data, depending on the different sciences and disciplines. So I have broken the first rule of making a slide by having a bunch of text on one, but I'll go through it quickly. Students, never make a slide like this. This is what professors tell you, right? Only 3 lines of text per slide, then they put up a slide that has 25 lines. Do as I say, not as I do. So in crops, and a lot of these have been mentioned throughout the last couple of days, the different types of data that we have access to, historical data, data that's generated in time, and forecasted data. Everything from remote sensing, satellite UAVs, terabytes of data generated daily, depending on what you're doing, what you're looking for. You have Internet of Things, in-field sensors, looking at soil physics and characteristics throughout the growing season, water measurements, in-field weather measurements, other characteristics of things happening in field. You have all the genomics databases and data generation that happens throughout a breeding pipeline. Everything from NCBI, which has millions and millions and millions and millions of data points or sequences, to specialized crop databases, MaizeDB, SoyDB, these other things that are very specialized around a specific crop or set of crops. And some of the databases for collecting phenotypes, which I don't think are as robust and as structured as the sequence databases for obvious reasons. You have public agricultural databases, give historical data on what crops have been grown where, what yields have been over time, other factors that go into that. Those are very important as you start building models to predict how things will perform, what agriculture is going to be like in the future. I'm really excited about the 5th one, and this has been going on for a couple of years. But the amount of data that we can collect on farm from machines in the field, everything from planting, what genotypes you're planting, how they're treated, what fungicide, herbicides are going on those, what the nitrogen, potassium rates are. So you can start building models that incorporate all this data on everything that goes into actual production in a grower's field, farmer's field, tied to a genotype or hybrid, whatever that you have going on there. This is John Deere's got incredible systems. The Climate Corporation, which is part of Bayer Corporation, has Climate Field View, which also generates a lot of data about in-field management and helps them actually predict and determine where and how to place different varieties. And then, of course, all the weather and climate data from various databases and in-season generation. So there's a ton of different data types. Like I said before, this is all very heterogeneous and very complex. This is not your typical physics data. If we think about it just from the genetics side, think about soybean, for instance. You have hundreds or thousands of genomes that you can access around the world that have been sequenced to various levels of completeness. You have hundreds to tens of thousands of RNA-seq experiments that have been done for different treatments, different environments, different genotypes. There's a lot of information there, a lot of heterogeneity, of course, and complexity in that. A bunch of other functional genomics that you can build on top of that, everything from bisulfite sequencing to ATAC-seq to contact maps and other data that you can start to build into this whole omics data set. And if you think about the genotyping that happens on an annual basis for soybean, and I'll talk a little bit about a particular breeding program, we're generating millions of genotypes per year. High resolution, low resolution. A lot of those are not accessible because they're done within corporations, but there's a lot of data being generated. And you can actually impute up to haplotypes into inferred sequence data. And then, of course, you can build all the relationships, pedigree and genomic relationships between these entities. So just think about a soybean breeding pipeline, just an average commercial soybean breeding pipeline, not the entire North America or entire Brazil breeding pipeline, just a segment, a maturity group. In any 1 year, you're probably making about 100,000 progeny. So 10 to the 5th F1 progeny. You're going to genotype those at low resolution. You have, if you don't recycle or reuse any parents in the crosses, you have potentially 1,000 different parents in those making those crosses, which is an overestimate because you're going to be using a single parent across multiple crosses. You have high resolution genotypes. You're calculating optimal contribution to figure out which parents to use, how big to build the populations. You're using the F1 progeny to impute back up to the high resolution and for genomic selection. Then you do purity testing, selfing. Then you get into testing. You're actually doing, testing, you're doing purity genotyping as well. If you're doing trait integration, whether it's a native trait or a biotech trait, you're also doing genotyping within that pipeline, which can double some of these numbers going down through there, all the way down to the point where you maybe release 10 varieties in a good year from that pipeline. So there's 120 to 200,000 genotypes per year per pipeline. And that's a lot of data generated just for one pipeline that you have to keep, you have to organize, you have to know the relationships. And that's correlated, or not correlated, but it's in addition to all the other data you're generating for all these different entities, their yield, their performance, how they respond to different environments and pests and pathogens. So it's an immense amount of data just from a single pipeline within a larger breeding program. So let me talk a little bit about AI. If there are any questions after the AI section, those go to Alex. So what is artificial intelligence? Basically, it's the idea you want machines to act like humans. You want them to think, create, do things that humans do. So think and act like a human, problem solve, decision making, generate creative content. It's rapidly, especially in the last couple of years, because of the generative AI and the large transformers, it's becoming rapidly ingrained in everything we do. Everything from Copilot filling out your letter or writing a letter for you or whatever to generating content through ChatGPT or Grok or whatever, to self-driving cars from Tesla or Waymo or BYD. And I think it's pretty incredible. This is data from Tesla. So in North America, the average accident rate is about I get this number wrong, 1 in 670,000 miles driven. If you use the auto driving feature in Tesla, that drops down to about 1 in 7.6 million miles driven. So there's a lot of benefit to these things as they become more abundant throughout our life. So the timeline, AI started back in 1950s. The term AI was first coined in Dartmouth, a conference in Dartmouth in 1956. Some key major milestones, machine learning, supervised machine learning is in the 1980s. This is things like taking images and predicting what was in the image, so a cat versus a dog, some of the SVM type things. Unsupervised learning, deep learning in the 2010s to where we are now in 2020 with generative AI and the large transformers that on the deep neural, large language models and deep neural nets that allow us to generate this original content. What's next? Who knows? This is going so fast. In the US, it's really only limited by our ability to generate electricity. So there's a rapid expansion in developing energy sources to fund these large data centers to run these AI models. But probably there's a movement from large language models. They're very powerful, very useful, to specialized language models. They have application in a very specific area. AI agents and swarms of agents, things acting in concert. Who knows when we'll achieve general intelligence? And there's various definitions on what that actually means, and Alex will fill you in on what he thinks it means. I'm putting all the hard questions to Alex, so he's right in the second row, front, right. So I just want to walk through a simple example of some work that we did in the lab before I went to Bayer, and I'll tie it in with some later work that when I came back from Bayer to academia. So thinking about tying a genotype to a phenotype, obviously the most direct route in a lot of genetics is relied on making mutations in a gene that lead to a structural change or stop codon that gives you some mutant phenotype. So you go from a gene, you have some non-synonymous SNP that leads to a direct phenotype, a change in a protein or a change in a plant phenotype through a change in a protein. But there's a lot of variation that happens through there's a lot of phenotypic variation that happens through changes in expression, and that can be done through EQTLs, so other genes that affect the expression of a gene, or SNPs upstream or changes upstream of a gene that affect the way transcription factors bind, and then lead to changes in expression that lead to changes in phenotype, or even epigenetic changes. And of course, GWAS, TWAS, a number of different tools have been used to try and explore to find genes that contribute to phenotypes. These have been somewhat limited. If you look at GWAS, you always get just a few number of peaks. There's a limitation in power. TWAS, I'm going to give you an example of some really nice TWAS work shortly. But the approach that we took was a machine learning approach. And we're focusing really on this, so changes in expression. So if we can find variation in expression patterns, and of course, if you think about variation in expression, you have some phenotype, you have some gene expression, plus or minus goes up or down, leads to phenotype. That can be through transregulation, this EQTL type thing. And this is a whole cascading thing. There can be multiple cascading effects that lead to a change in a gene expression. And you don't capture this through a GWAS. So the approach we took is we took some public data from Mays, this is from Ed Buckler's lab, and from Common Bean. This is the GWAS populations. The Mays one is 200 individuals. The Common Bean one from Phil McLean at North Dakota was 100, and I'm going to talk about the Common Bean one here. They'd measured phenotypes, but they'd also in addition to genotyping and to GWAS, they generated RNA-seq data across the entire population. So in the case of Common Bean, they collected RNA-seq data at the 3-leaf stage. This is very early in development, juvenile stage. So that's where the RNA-seq data is collected. And we used just a very traditional, and we were very naive approaching this, very traditional machine learning approach to look at variation in expression of genes across the population. So we're looking at genes that are varying in expression across the population of 100 or 200 individuals to try and build a model to, one, predict the phenotype, and two, to get better insight into the genes that underlie that particular phenotype. And so we used ridge regression and SVM. And what you can see is we generated GBLUP predictions for these. This is Common Bean, so we had 100 individuals. After cleaning up data, we were looking at about 11,000 genes. So the genes had to have some variation in expression and not hypercorrelated. And so there's 11,000 genes that went into our model running through ridge regression, SVR. And what you can see in black is what you get from GBLUP predictions. And using variation expression, so this is using variation expression to build a ridge regression model to predict the performance of some holdout group from your when you run the model. And you're getting predictions that are as good, and in a few cases better than GBLUP. So yes, we can predict phenotype from variation gene expression using this model. We can also, you can do a recursive feature elimination, so you take the lowest ranked feature out of the model, run the model again, do that again, until you're out of features. And then you have a ranked feature contribution to that model. And what you can do is you can go in there, use gene ontologies, or just look for genes that you expect to be there. And looking at flowering time, we found enrichment for genes in the gene ontology groups that you expect, but just looking at like the two integrators in flowering time regulation, FT and FST, or TSF, so flowering locus T and TSF, there's 2 copies of these in Common Bean. We found both of them, and 3 of them in the top 155 genes. One of them was in the top 1,500 genes. I don't know where to make a statistical cutoff in this recursive feature elimination, but if you just think the top couple hundred genes that contribute to the model. And what's interesting is none of these were found by GWAS. The very top one was found through TWAS, but the other ones were not found through a TWAS approach either. So this is not only can we predict the phenotype from variation in gene expression from juvenile collected RNA-seq data, but we can also begin to get a better understanding of the genes that contribute through variation of expression to those various phenotypes. So these are gene candidates for breeding to build into selection models and for genetic engineering. I'll come back to that. We also see that gene expression is highly heritable. across populations. We actually go outside of this population and common mean and predict in other populations, and we can rank them in order. We can't predict the actual phenotype, but we can rank the phenotypic order. You can predict adult phenotypes from juvenile samples. I was a bit surprised at that. Even flowering time, things that you sort of I naively did not expect to be able to predict. And many candidates are acting in trans, which you sort of expect. And I want to point you to this paper from James. I don't know if he's going to talk about it at all, because I think this is a really beautiful, using TWAS to get an even deeper understanding into the gene's underlying traits. And I think this is really powerful. This is published a year ago, I think, or last year. But I'm not going to talk about it. Hopefully he talks about it. But it's really, all the students should read this paper. It's a really beautiful paper. All right, so I want to end this section with some caveats. Big data does not equal big insights. And this is something that I'd been ruminating in my mind for a couple years, and I had a colleague at Bayer, I'm going to mention him later. He comes out of physics, and there's all this, and it's not just industry, it's academia, it's everywhere. You know, generate all this data, we're going to suddenly have insights, profound insights that we never had before. And that's not the case. Most times it's not the case. I would say most of the time it's not the case. Data is abundant, but novelty is rare. AI is very good at interpolating and predicting. It's not good at creating and generating new knowledge. And I really have to give credit, because I'd been thinking about these things. I did not articulate them this well. I actually pulled this from a talk that was given last week at Harvard by Marcus Buehler, who's a professor at MIT. I highly recommend that students, whatever, that QR code will take you to that talk. It's a great talk on where AI is going. He's a material scientist, but where AI is going in research, how they're using it, how they're building swarms of AI agents to solve problems. But he's also very clear that this is it's not generating hypotheses. You have to pose hypotheses. You can tell it what to do. You can put the hypothesis in front of it, but you really have to tell it what to do. So it's really good. Big data does not equal big insights. We have abundant data, but novelty is rare. And AI is poor at creating and generating new knowledge. Go watch that talk. All right, let me try and pull these three things together and what we're trying to do now at Georgia. So I think we're really in a time of an era where it can be disruptive in how we think about breeding. We have all these rapid advances going back, the genomics revolution goes back to the 1980s, 1990s, but it's really only the last 10 years that's become really democratized and much less expensive. And I put that in quotes because inexpensive, depending on what you're generating, how much it costs, and is relative. But it's become much more democratized and much less expensive than it was, a decade ago. So the ability to generate sequences or RNA-seq data or any omics data or genotype is almost trivial at this point in many cases. Gene, and I put genome editing. There's still a lot of work to do on some of the technology and techniques, but it's really a revolution that can change how we think about breeding. So the ability to edit specific genes to have different expression patterns, or what I call genome editing, to manipulate multiple genes at one time to create new variation within a breeding population or to create new phenotypes that you want to select upon. And of course, transgenesis has been around for a long time, but it's becoming more democratized. We had the talk from Corteva where they've allowed their transformation technology to be used in academia. That's really advanced a lot of what we can do in academia. So that's really moved the field forward. I talked about the AI and machine learning, so all the advanced, the large language models, the specialized language models, all the deep learning that we have recently, the access to high computing GPUs at a I'll put really in quotes, relatively inexpensive. We're in the process of buying some H200s. It's about $300,000 for 6 of them, for a stack. And then synthetic biology. Synthetic biology has been around for a long time. But it's been mostly in microbes. So engineering new biosynthetic pathways or new molecules in microbes. It's only really in the last 5-10 years that we're starting to see traction in plants on the ability to build complete biosynthetic modules that you can potentially move into plant to create an entirely novel biomolecule that might have potential value. Still a lot of work to do, but I think it's very promising. So how do these things come together? I apologize to all the breeders in here, and I'm trained as a plant breeder. This is how I think about breeding. You can complain to me afterwards. I don't know if the analogy of searching for a needle in the haystack translates in Portuguese, but you have a giant stack of straw, and there's one needle in there that you're trying to find. And to me, that's plant breeding. You start these massive populations, right? And you're looking for that one thing that's going to perform really well across a broad range of environments. And so you go from massive population sizes, few number of environments, over time, looking for those few needles in that haystack, and then increase the number of environments that you're testing in to make sure they actually work. Again, I apologize to all the plant breeders in here. So can we be more design-driven in how we think about this? And I think this is not new. We've been doing this for a long time. A lot of the quantitative models that we've used for the past decade, 20 years, are really driving that in that direction of being more design and how you build populations, how you select parents, and how you move things, how you test things throughout a pipeline to one, increase the frequency of needles, or to reduce the pile of straw so that you don't have to search as hard to find those needles. And so can we expedite breeding? Can we think about recycling parents more quickly because we can predict how they'll perform even though they've not been tested for many years or not tested at all, and increase the heritability because we're reducing that time component. And there's a lot of building omics data into that. There's a lot of modeling. There's a lot of quantitative genetics that goes into that. Like I said, can we decrease the size of the pipeline? Can we increase the frequency of needles? We're seeing this already. This is happening in academia and industry already, but being much more intentional in how we build the populations at the beginning based on what we expect the performance of the progeny from those parents to be. So not making everything, 100 crosses, each population is 200. Now it's like, we think this population is going to be really good. We can only afford these many things in the F1, F2 stage of testing. We're going to put a heavy emphasis on this population, and maybe it's 20 or 30% of our entire testing footprint for that first generation because we think this is a winning population. So it can be much more intentional in how we build those populations going through a breeding pipeline. Can we, from the very beginning, engineer more precisely for target environments? And this is happening, I would say, there's a lot that can be done to improve it, but it's already happening in some of the bioeconomic indices. So understanding what your target environments are going to be, what the management styles are going to be, all the different environmental pressures, and building selection indices all the way back up your pipeline that put weights on how to perform or what the economic trade-offs are in those target environments and management styles. There's a lot of work that can be done to improve those, but they're already being used. And you can reach sustainability goals using this. Using the synthetic biology approach, can we use more of the crop? Can we increase the value? So if you're harvesting grain off a field, of course you want to leave stover on the field to keep soil fertility, but are there additional things that we can put into that that'll give value to the grower? Are there biomolecules that'll have high value that they can extract from leaves or some part of the stover that would give additional value to the growers and give additional profit margins? So the approach that we're using, and this is this design, build, test, learn cycle. And it's been around for a long time. This is an engineering approach this is an engineering cycle. But even if you think about breeding, this is the breeding cycle, right? You design, build populations, you test them, some get kicked out as varieties, and you learn from that, and you run the cycle over and over and over. What we're trying to bring into that is all the omics on the learn and design side. So can we bring in all the information we have on pedigree relationships, performance, optimal contribution, AI models to better understand G by E predictions of progeny and parents, to design and build those populations, or design and build new pathways, or generate new variation within populations that we think will be beneficial within that breeding pipeline. Can we build and test in vitro? Can we run the cycle more quickly because we can build and test and predict performance in vitro, either through cycling in culture or cycling in protected culture before we even kick anything out to the field for testing where it's more expensive? And so can we run this cycle much more faster? Can we be more informed in how we do the various points throughout the cycle and help the breeders to really meet the goals and be much more expedited in how they run breeding pipelines? Expeditious, I should say. So the way that I'm conceptualizing some of this, and this is courtesy of Ethan Pickering from Bayer. He and I have been, for a couple of years now, he's a physicist that we hired at Bayer when I was still at Bayer. Works in the artificial intelligence and modeling area. And thinking about the different scales and all the data that we generate and the data that we have access to. Everything from the nanometer level, the genomics, the genetics information of the plants and the populations, to the meter level, to the phenotypes of a plant or of a plot. all the way to the kilometer level of understanding the soil landscapes and the weather landscapes and across those different scales, bring all these things together to build prediction models that allow you to be better informed in using quantitative genetics and AI and machine learning to build G by E models to bring all this information together, bioinformed networks, to predict performance of something within a different landscape. And that helps all the way from the beginning of the pipeline, from how you build populations, understanding what the landscape is that you're going to be putting it in, the management styles, all the way to how you do your testing, how you build simulation models at the phenomic level. You have real data, but you can also build synthetic data to build simulation models that allow you to much more deeply understand the genotypes what you're putting through there and how they might perform, to understanding where to place them for testing, where to put testing sites to get insights that you don't have that you need to build the models. So maybe it won't perform, but you don't have that data to understand that it's not going to perform in some certain areas. To collect that data so you can help build and strengthen these models. So really linking across all these scales to really bring the physics, the AI, the quantitative genetics, all the sampling, and all these things together to be much more predictive on running the entire breeding pipeline. So let me come back to bring this down to a lower resolution. So we've talked at a high level about running these breeding pipelines, collecting all these data at these different levels, these different scales, building bioinformed genetic networks that allow you to use digital twins to simulate phenotype and performance in various environments along with real data, to understanding how they're going to perform in different environments and do predicted placement and testing. If we come back to we want to build new variation or new molecules within a plant or a crop. I talked about connecting the genotype to phenotype. You know, changes in genes that lead to a phenotype, or you have changes in expression. And this is my simplified breeder's equation. So response is heritability, selection over time, selection index or selection intensity over time. So we have goals of increasing heritability by understanding the underlying genes. And we want to identify additional genes for engineering new variation within those pathways. And we want to potentially engineer completely new pathways. I presented this common bean data. This is the same data for corn. And you can see that ridge regression, SPM also perform as well as GBLUP and better for a few of the phenotypes. So we can use the same thing for corn to get a better understanding of the genes that underlie various traits to help build into these prediction models to increase the heritability. And on the engineering new variation, I think this is a really exciting area. So this is actually a figure from Tom Jacobs and Dirk Inze from VIB in Belgium, where they multiplexed editing targets within corn to change expression or change gene patterns in multiple genes, and then make crosses, then they could predict crosses to make among those progeny to add together all these different genes that you want to change expression patterns and generate new variation in. And I think this is what you can begin to think about as you understand the genes that underlie a trait, and you can use these different machine learning or TWAS approaches to understand these genes. Some genes may not have variation that you can select on. So this is a way to engineer new variation. Or if you don't have sufficient variation, this is a way to engineer new variation within a population. You can use this to build new quantitative models. Or if you can really precisely understand, I'm going to move this gene in this direction, it's going to result in this phenotype, then you can do it in a much more predictive manner. And that gets really complicated. So if you think about a really simple gene network, gene 1, gene 2, gene 3, we're interested in protein 3 coming out of gene 3. So you can alter regulation of gene 1, gene 2, because they both do that. Let's just pick gene 1, because it alters regulation of these other two genes. So this is really simple. This is just 3 genes. How do you do that? How do you know how to go into promoter sequence or other transcription factors that affect that gene and very precisely alter the regulation or transcription of those genes in a very precise manner, very cell type, very tissue-directed manner, and very modulated? So CRISPR right now, if you want to change the expression of a gene, it's very good at changing the expression. It's very poor at predicting what that change in expression is going to be. It's basically like old school promoter bashing. And so you can get a lot of variation. You get up, down, you get all sorts, or turned off completely. But if you want to go in and precisely engineer a gene to go to increase transcription by 10% only in this cell type, that is really complicated and really difficult. And that's one of the things that we wanted to start exploring. And so this is a new postdoc, Olive Fargaden, and Ethan is co-advising. And I should point out that all this data comes from Bob Schmitz, I forgot to put his name on here, a colleague at University of Georgia. And what we want to do is see if we can naively go through the soybean genome and use deep learning models to try to use DNA syntax, DNA language to understand how promoters and areas around transcription start sites affect the regulation of that gene. So the approaches we took all the genes in soybean, we took plus or minus 500 base pairs of the transcription start site, so 38,000 genes. We used single cell RNA-seq, so this comes from 7 different tissues. We have 79 different cell types that are represented in there. At the very first, we just took a pre-trained genomic pre-trained network, and this is actually done in Arabidopsis. This was published. I think this is an old slide deck. There was a reference in here for the paper. It's 2023 PNAS. And you could get mean overall prediction accuracy of about 0.45. If you use the EVO2 models, which are not trained, they're trained across the entire NCBI basically. It's very close to what you get for an Arabidopsis model. If you fine-tune that the GPN model for soybean, then we can get like a 30% increase in performance relative to the one that's just trained in Arabidopsis. So there's a lot of value in taking these models. So we're getting back to that specialized language models, making sure they're adapted and trained on the data that you want it to be used for. So we use that as the input for a convolutional neural network. So that was the input G, the output of this GPN. We have the cell type embedding, which comes from these different cell types up here, the 79 cell types. We ran it through regression heads with learnable weights, and we tried to predict the output, gene expression. And so this is going to go very quickly, but what we did is, so I talked about interpolation. So these are basically interpolation up here. So if you use 95% of the data to train and you test you do 5% holdout, and this is a random holdout. Train on 50%, do a 50% holdout. Train on 95% and try and predict on 5%. So green, blue, red. This is the Pearson correlations on the top, and this is the MCE, the mean squared error on the bottom. So you want high PCCs and low MSEs. So the blue and the green, the 5% and 50% training, or 95% training, 50% training, you get Pearson correlations up above 0.9. When you go to the one where you're using you train on 5%, predict on 95%, then it drops significantly down to 0.7, although it does go up in certain tissues. The open triangles are if you only train on a tissue. So the closed ones, we train on the entire 79 tissues, or 79 cell types. The open triangles and circles and boxes, you can't see those because they're superimposed in blue and green, is when you only train on a certain tissue. So there might be 7, 10, 12, 13 cell types in that tissue. And there the correlation drops and the MSE goes up. So those are pretty good. When he showed this data like 0.9, 0.95 prediction, like this is awesome. We can now begin to think about how to dig deeper into these promoter regions, these TSS regions, and understand which parts of those regions are contributing to the control, the cell-specific expression of those genes. Not so fast. So I'm just showing the data from the training on 95%, predicting on 5%. So this is like the best. Here's the Pearson correlation. And what we're looking at now is the number of cell types it's expressed in. So this is only one tissue. So we have 13 cell types in this tissue along the X. And what you see is you have really high Pearson correlation in blue for genes that are not expressed at all, never turned on. And you have really high Pearson correlation for genes that are turned on all the time, housekeeping genes. So that's what's driving those correlations. Most of the genes are either off or on. There's very few genes that are actually have cell-specific modulation or turning on and off. And this model is really poor at learning those. So this is what's driving those correlations. That's where we want to be, and that's where we have almost no correlation. We have really high mean squared errors. So we can predict, but what is really those 0.9 whatever Pearson correlations, it's really predicting on the gene ID. And it's going, if you hold out 50% of the data, but this gene is on in the other 50%, the model is just saying this gene is always on, and those 5 is probably on in these 5. It's not doing anything with the TSS. So learning syntax for cell-specific expression is actually really difficult. That's the takeaway. And he's only been doing this for about 6 weeks. He's a physicist, so this was his first project to understand what DNA is. But I'm still enthusiastic. I mean, I gave you this We're going to learn the syntax of how to control these genes in a very specific manner so we can engineer pathways and so forth. It turns out to not be that easy, but I'm really still excited about the future. And this is a paper from Xie et al. I think it's still pre-publication status for Cell. And if you think about from the design process, what they're trying to do is design, this is tomato and soybean. They're trying to design soybean and tomato so that they can rapidly cycle them using robotic breeding. So in order to do that, if you think about soybean, anyone's ever made a soybean cross, soybean is cleistogamous. It wants to self before the flower even opens. So you have to know how big the bud is to dissect it to make crosses between soybean. Otherwise, it's going to self itself. And it's really difficult to think about developing robots. They're going to go through, this is the bud I want. It's going to emasculate it and then make the crosses. So the approach they took is like, let's engineer the flower so that we can then engineer the robots to make the pollinations for us. So using the information that we know about how flower morphology develops, they're able to go through for tomato and soy, both explore male sterility, but also create exerted stigma. So that the robots, these exerted stigma before it self-pollinates, or they have a male sterility on top of that. So then these robots can go through, apply pollen to those stigma, and they can do rapid cycling, rapid breeding within a protected culture. I think that's really cool. There's a lot of promise. If you think about developing potentially hybrid soy, this is like the first step. You have to have flowers that are open for insect pollination or some manual pollination, and then engineering male sterility on top of it. So really cool. One last example of one thing that we've begun work on at Georgia, and I picked this example. We don't have any data on it yet. This is something we're still working on with some engineers, is peanut shell. So about 30% of the peanut is shell, and that ends up in waste streams. And the reason I picked this example is because peanut production in Sao Paulo, this state, is on the rise. I think it's 1.2 metric tons or something predicted this year, which is about 50% of what the US produces, and you guys are rapidly going up. So in the US, it's about 1 to 2 million tons of shells are produced every year. This mostly goes into waste streams. Some of it goes into low-quality animal feed, but for a number of years, people are trying to figure out, what do you do with this? You have a couple million tons of shell. It's not really good to put back. It doesn't do much for soil fertility to put it back on the fields, so it mostly goes into waste. But they've looked at, can we extract the fibers from it to strengthen concrete? And it's had a little bit of value, but the cost to extract the fibers from it does not offset the value to the concrete production, so the ROI is low. They've tried using it for biochar. It doesn't work terribly well for that. So the approach we're taking is, what can we engineer that will provide additional value for the grower or the sheller or the entire stream of peanut use? And so we're working with some engineers and some people at the Carbohydrate Research Center at University of Georgia to understand what the composition of the shell is, what the potential valuable or what potential molecules that we might engineer to be more valuable, or new molecules that might be engineered in there, or what we can change to make things more extractable that might be more efficient to extract the fibers for construction material. So that's what we're working on now, one of the projects we're working on now, and using this whole test of what do we know, what can we learn, what can we design, how do we build that, and how do we test it. And one of the caveats in this is, nowhere in the world do they want GM peanut. So that's one of the things we got to figure out how to deal with. So it's probably going to be some editing approach. We have to get rid of all the editing machinery and everything else. But it might be much simpler to actually engineer a new pathway or new molecules just by putting in new genes. But no one's going to buy a transgenic peanut. If you go to the grocery store in the U.S., almost every jar of peanut butter says non-GM. All right, so let me close up here. And I think there's a lot of opportunities and challenges. And I think it's a really exciting time to be a student and postdoc in this field. It's scary for an old person like me because it's moving so fast and the technology changes so quickly. But I think it's a really exciting time because there's so much happening, there's so much opportunity. And there's so much opportunity to bring people from different disciplines together to solve really complicated and what we thought were intractable problems. So we have challenges. We'll always have challenges. And I'm not talking about food production challenges, developing parts of the world. I'm just thinking about technical, scientific challenges. We have, there's still challenges about access to data. And some of this is, companies generate a lot of data, and that data would be very useful for academic research, but it's very difficult to figure out relationships where you can make that sharing possible without revealing secrets or things that the company wants to keep confidential. I think that's something we have to continue to work on, but it's a challenge right now because, at least for the major crops, corn, soy, cotton, wheat, canola, those are mostly coming from private industry. And how do we as academic partners work with them, work with their data to actually enable and produce new things? I think it's a challenge, but I think it's something that we can make some solutions. Data asymmetry, I'm gonna give you a real easy example of data asymmetry. If you think about a breeding pipeline, I showed you start with a large number of genotypes. By the time you get down to the end, you have very few genotypes. You generate very little data at the beginning. You're doing very little testing in all these genotypes. So you have all this genetic information. The genetic area is very big. The phenotypic area is very small. You get down to the end, your genotypic area is minuscule, but your phenotypic data is huge. So there's asymmetry between the data you generate and the genetic area that you explore. And it exists for that principle exists for other things as well. There's a lot of proprietary technologies that block access. It's awesome that the CRISPR-Cas and some of the transformation technology for Corteva has been enabled for academic research, but there's a lot of other technologies that are blocked and are difficult to use. Model biases, that's existed forever. But I think there's a lot of opportunities. Gene editing is a routine breeding tool. I'm really excited about the potential there. So if you can inject variation into a breeding population or pipeline for a specific output or phenotype, and you can do that in a very prescriptive manner and understand what it's going to do or build it into your quantitative models, I think that's really exciting. A lot of potential to optimize testing strategies, think about how and where you test to get the information you need, but also to be much more prescriptive, to get the data that's going to allow a variety to be successful, and you can give that information to growers that this variety is going to perform well under these certain environment and conditions. A lot of work to improve genotype to phenotype models. A lot of work to happen there. And I'm really excited about going back to that first thing, access to data. I think there's a large opportunity to increase public-private partnerships and collaboration. All right. So, a big thanks to the organizing committee. The one in the middle is looking at the sky for some reason. Where is Victoria, right? Where is she? Is she in here? I wonder what was up on the ceiling. But like I said before, I've been here several times to Brazil. This is the best visit by far, and it's because of the students that put all this together and all the work. They've been driving us everywhere, making sure we don't get lost. They don't want us walking by ourselves. Where's Joe? But just fantastic, and a big thanks and congratulations to this organizing committee. One of the best organized symposium I've ever been to in my life, so congratulations. All right, and then to thank my collaborators, Ethan Pickering is at Bayer, Ollie, the postdoc, Chun Ming, who did the machine learning work. He's now faculty in China. I'm starting a new institute. We're looking for faculty. We're looking for postdocs. So if you're interested in those various things, feel free to reach out. And I should have put Bob Schmitz up there, but hopefully he understands and never sees this. So thank you.
Dr. Jackson, on behalf of the organization committee, would like to express our gratitude for your insightful lecture. As a token of our appreciation, would like to offer you a small gift.
I'm just going to shout at you.
Yeah, I'll repeat it.
All right. So I really appreciated the whole talk, but particularly talking about the challenges of how you can get really good-looking predictions for expression from promoters when you're not really predicting from promoters.
Yeah.
But the big challenge, we only have 30 or 40,000 genes for any given species. How do we get around that? If we want to train these models, how do we get enough data to train something that really was going to understand the language of promoters?
Yeah, so he's asking, how do we get enough data to actually train these models to understand the promoters? These genes are only expressed in very specific locations. So in some of those tissues, like the genes only expressed in one cell are like 70 genes. So it's really difficult to train a model at 70 genes, and especially when they don't use the same syntax. So we're introducing some loss functions into the high and the low and the housekeeping, the always on and always off genes. But I still don't think that's going to solve the problem completely. So you have to have probably more samples, more genotypes. So we're going to look at pangenomes and RNA-seq data that have been collected for those. I don't know if that's going to solve the problem, though. Ask me in a year. It's a really, really challenging problem.
So throughout this week, we've talked about all of these technologies that were promised to be revolutionary and have ended up having relatively little impact on plant breeding. How do we take a technology with such great promise for improving the breeding program, particularly for increasing the number of needles in the haystack, which is probably one of the most important things we could do to make our breeding programs more efficient? How do we make this technology, what do we need to do to increase the likelihood that this technology actually does become embedded in almost every breeding program?
Yeah. Well, I'll answer the second part, how you embed it. The first part's much more difficult. So I'll take the easy part. I think, so what we're trying to do, this is a standalone institute, but we have an institute for plant breeding at the University of Georgia as well. And what we're trying to do is be very intentional in how we partner with them, both individually with individual breeding programs, but also with their students in training to try and make this a part of a collaborative effort in both how we train students for the future, but also how we do research currently. So we're working with like Zhenglu Li, who's the soybean breeder. We actually have a shared postdoc that's trying to introduce some AI models into what their genomic prediction, but also their testing pipeline. And he's working with this group as well. So we're trying to like build that cross pollination, collaboration, connection, whatever word you want to put in there. I don't know when the return is going to happen, how soon it will happen, how big the return will be, but we're trying to be very intentional in building that connection right now.
I'll let you think about that for a week, and I'll
Ask me about after 3 Caipirinhas tonight. I have a very loquacious answer.
Scott, we have seen, at least here in Brazil, a lot of attempts on using, let's say, AI for genomic prediction, for example. So we've seen, for instance, a lot of researching applying shallow networks and considering deep learning. At the same time, we've seen a lot of naive attempts on doing a lot of stuff to gather better predictions. But we're seeing how AI is growing so fast, and we're seeing the need of a lot of data. But we don't have this data, because usually the experiments and the investigations that we have, at least here in Brazil, they are very tailored to a specific crop, to a specific scenario. So usually the best is always the same approach. So what would be your advice for new students that are starting to develop a project in a world that is evolving so fast regarding AI, but they don't have data for taking advantage of these new technologies? What do you think they could do, to at least try to incorporate some kind of attempts on improving what they are doing, or at least have an idea of what could be the future like which kind of data sets, what kind of integrative approaches they could do? What's your opinion and suggestion on that for the new students?
Yeah, so I have a lot of answers to that one question. And many of them are context dependent. One, so a student that's really interested in machine learning, AI, whatever, and doesn't have access to that data, but can do an internship and go work at Corteva or Syngenta or GDM or whatever, and have access to that data and work along their scientists and see how it's applied and how it's used is one. But they've already built that experience. So they're not going in there for a 6-month internship and have no idea what an AI model is or anything. Another one would be to generate artificial data at the scale you need, synthetic data. And that's done, physics does that all the time. Their experiments are so expensive, they can't afford to run all the iterative experiments that we do in breeding. So it's all synthetic, and then they run one to confirm it at the end. So synthetic data. Bayer, I don't know if other seed companies do as well, but they run data hackathons. So they've actually taken actual data from breeding pipelines. It's anonymized, and they actually give it to the university. And we just ran a hackathon 2 weeks ago. There are 2 Brazilian students from this university that are up there now, and we're part of the hackathon. And they leave the data with us, and they can use that to experiment with. That was 3. I had a 4th in my mind, but I've already forgotten what it was. But,
No, that's fine. Thank you.
Thank you. Congrats for your talk. As a soybean breeder, I'm very happy when you said about the hybrids in soybean. Can you share a little more in your vision? How can I use this in the future, if in your opinion is possible?
To create hybrid soy as a yeah, okay. So I get enthusiastic about this every couple of years. It's sort of like hybrid wheat. It goes through cycles where companies they build hybrid wheat breeding programs, and then they realize that the cost of goods is too expensive, and then they walk away. I remember I was at a World Soybean Congress in China 15 years ago or something. Maybe you were there. And there was an older Chinese soybean breeder who had worked on hybrid soybean his entire career. And I actually visited his fields. I think he was up in Jilin, so northern. And he had built all these cages to try and force insect pollination. They'd been selecting for more open flower characteristics. And basically his entire talk was he spent 30, 40 years working on hybrid soy. You could get, I think, 15, 20, 25% a boost in the hybrids. But his final slide was something to the effect that this will never work. So I'm going to start with that. There's a number of projects now working on engineering soy flower morphology. So that one that's in Cell. There's a group at Cornell. Bayer just announced they have an initiative with a public group, actually, at the University of Georgia, to introduce apomixis into soy and re-engineering flower morphology. So to me, that's the first step. So if you can engineer flower morphology and if you can engineer male sterility, that's the first step towards creating hybrid soy. And there's a lot of details that go into both those and that come out of it. What I still don't know, and we might run into the same problem that wheat has, is that so few seeds are produced per cross that the cost of goods is still prohibitive to being a useful technology. And that I don't know. I'm still excited about the opportunity. Even if you don't get to hybrid soy, if you can engineer the soybean flower so you can make crosses more easily, or you can even have robots do it, to me that's a huge, anyone that's ever made a soybean cross, that'd be like a massive step forward, right? But I don't know if the cost of goods ever allow us, at least where you get one, 2, or maybe 3 seeds per cross, get us to the ability to have it as a viable hybrid crop. That's my big concern.
Again, I would like to thank you, Doctor, for his brilliant presentation.